Microbiology by Laureano Andrade Vicente and the MCAT disciples. So here's the AMC topic list that we're going to go through. The only thing on here that I don't have much to say about is the prions, and I'm going to mention them, but I'm not really going to dedicate a slide to them, so that's why I crossed it out, but I'm going to mention them. They require a little bit more of an understanding of protein folding and the hydrophobic effect, but I'm going to mention it very quickly, but I'm not going to make a big stink about it. Okay, so we'll start simple with the relative size of biological entities. You don't really need to know any of the numbers here. Just like it says, you need to know relative size. You know that most plant cells are larger than animal cells. Most animal cells are much larger than bacteria. Most viruses are much smaller than bacteria. And most molecules, of course, are the building blocks of everything, are going to be the smallest. Then atoms. The relative notion. Now, again, these pictures aren't necessarily very accurate in terms of relative size, because many viruses can fit into a bacteria. It should be a lot smaller than this. But just to get you an idea. So let's continue. Now, there are the two broadest categories of organisms that exist are the prokaryotes and the eukaryotes. The eukaryotes are just lumped up together. There are no further subdivisions in terms of the eukaryotes. But the prokaryotes are divided into the bacteria and the archaea. So that's what we see right here. And what we're looking at right here, the phylogenetic tree of life. We're looking at a common ancestor that then diverged into all these different organisms. Every time you see a node, it's a divergent. So here's the common ancestor here. Here's the common ancestor here between Thermotaga and green filamentous bacteria. So that's how you read that. And if you increase the length, you increase the difference in genetics, the difference in sequence of the genome, and also time. So it represents both of those things, the distance of the lines. All right, now this thing is based on ribosomal RNA data. So we took ribosomal RNA data from all these bacteria and mapped it and compared them and made this phylogenetic tree of life, okay, based on differences in the genome. And we use ribosomal RNA. Why? The ribosome is an integral part of every single cell that is alive. Every single cell that is alive has a ribosome. There are some slight differences between the ribosomes of every domain and every species, and that is one it allows for us to use antibiotics on ribosomal bacterial ribosomes, but it also differentiates the different domains in part. And so we can use ribosomal RNA data, RNA that works within a ribosome, to then calculate these differences and compare these differences. And we can do that by comparing their genomes of ribosomal RNA. And you should appreciate the fact that since the ribosome is an integral part of every single cell, then having mutations in the ribosome is generally unfavorable. Unless you get a gain of function mutation, it's generally unfavorable, because if you get a mutation, it's going to be a loss of function mutation. And a loss of function mutation is going to lead to less of an ability to compete with the surrounding environment, with the surrounding organism within the surrounding environment. So whenever you get a mutation in ribosomal RNA, it was probably a speciation event if you survived, okay? It probably created some new form of that species, or it created a new species. And that would be then an indication of the genetic distance between one species as compared to another. And that's what allowed us to calculate this map, because then they can say, well, this aquafox has 3 polymorphisms that this common ancestor doesn't have. So that's a new species, right? So that's how that works. But the question really is, why would we use ribosomal RNA data as compared to something else? Well, I just told you, essentially. Ribosomal RNA is resistant to mutation. It's resistant to mutation because any mutation you get, it's usually deadly, and the bacteria can no longer compete and they die. So that's how natural selection works, right? They naturally select for that, for the ribosome, for the bacteria that can compete the best. And those bacteria are usually bacteria who don't have mutations within their ribosome. And the other good reason is that within a species, right? You can have picked 100 different species of aquafox. I mean, you could have picked 100 different organisms that are of the species Aquifex, right? And any one of those species can have mutations within their genome that are just random, that they gained within their lifetime. That's not indicative of the entire species. However, in ribosomal RNA, you are much less likely to pick out an organism within a species that is not reflective of the entire population. And that is because the ribosomal RNA is very difficult to mutate. It's very resistant to mutation because of what we discussed earlier, because of this notion of natural selection and having, if you get a mutation within the ribosomal RNA sequence, you're probably going to die. And so that's why we use ribosomal RNA data, because the data that you get is very likely to be reflective of the entire population. And I also want you to appreciate the fact that the ribosomes between the prokaryote and the eukaryotes are very similar, but they are different enough such that we can design molecules that can fit into the different active sites of the ribosome that can diminish ribosomal translation within prokaryotes, but not affect the ribosomal translation in eukaryotes. And that is very effective for then killing bacteria in our body, because then we can take in antibiotics and eat antibiotics that are going to inhibit translation of the prokaryotes, but not inhibit the eukaryotic translation, which is a great thing. Anyway, that's not really important. What is important is differentiating between bacteria and archaea. So the bacteria and archaea are very similar. I mean, they're both prokaryotes, but there are some differences. But I'm not going to make a big stink about it, and the MCAT won't either. The one big difference that you should know to differentiate between archaea and bacteria is that archaea many of them live in extreme environments, either very hot or very cold. That's the one big difference you should know. But as you'll come to appreciate in the next slide, they're very similar. So prokaryotes, bacteria and archaea. So this whole conversation, I'm gonna often say bacteria, but I'm also talking about archaea. But I'll probably say bacteria the most, because we're really looking at bacteria for the rest of this lecture, not about archaea. But anyway, all right. So here's the general picture of a prokaryote with many possible appendages and variations. So this is actually an example of a gram-positive bacterium. How do I know it's gram-positive? Because it has cell wall and a plasma membrane. It's actually encapsulated as well, so it's an encapsulated gram-positive bacteria. Let's note some of we have pili here, we have ribosomes, we have plasmids. We have bacterial flagellum, and we have a nucleoid, a circular DNA. Notice that there is no membrane-bound organelles. You don't see an ER here. You don't see a nuclear membrane. You don't see no nucleus here. There's no nucleus here. There is no nucleus here. So this chromosome is just free in the cytosol, and this is one big chromosome. This is one big tangle of chromosomes. There's no more than one. These cells are haploid. Okay, they might have plasmids that may make them partial diploids, but by and large they have one chromosome, and this is it. It's one big tangle, one big gamish, and they have ribosomes. But that's about it in terms of organelles. That's about it. Ribosomes, and they have the chromatin. But that's it. That's it. So, and that's the general picture. That's what you need to know. So the next thing we're going to go through is, well, just different microbiology stuff that you should know. So we're going to deal with bacteria from now on. All organisms have a name consisting of two parts, the genus followed by the species. So Escherichia coli or E. coli would be how you would call Escherichia coli. Notice it's italicized, and notice that the species is lowercase while the genus is always uppercase. And you can abbreviate it by putting just the first letter. So the mnemonic goes, King Philip came over from Germany swimming. So it goes kingdom, phylum, class, order, family, genus, species. King Philip came over from Germany swimming. And domain is obviously at the top, because I told you earlier it's the most general classification of organisms. Just mentioning it though, you don't really have to memorize that anymore. What you should know is genus and species. They're the last two. Most important, really. Those are the taxonomical classifications of organisms. Bacteria are further grouped or divided and named primarily based on their morphological or structural and biochemical or metabolic differences. So one of the most common classifications is the gram stain. That's one of the common classifications we use, and that splits microbes into two categories. That's gram-positive or gram-negative, okay? And it also allows for determination of shape, okay, because it's hard to see without the stain. Because again, bacteria on their own are colorless. They don't have any color, so we need to stain them in order to visualize them under a microscope. The staining process goes fixation, you wash with water and you crystal violet stain them, then you wash with water, you add iodine, you decolorize, and you counterstain with safranin, it's a red dye. So then if they took up the blue dye, okay, they're gram-positive. Okay, or the crystal violet dye, they're about gram-positive. So the mnemonic is, I'm positively blue over you. If they didn't take up the blue dye, but they took up the safranin red dye, then they're gram-negative. Okay, and the different stains are results of the differences in the cell wall of gram-positive and gram-negative bacteria, which we're going to go through next. But you should appreciate the fact that, and you don't have to understand how exactly it happens, you just have to appreciate the fact that it's due to the differences in the cell wall that one cell takes up the gram-positive stain or the crystal violet stain, and the other cell doesn't take up the stain but takes up the safranin stain. So both gram-positive and gram-negative organisms have more than one layer protecting the cytoplasm and the nucleus from the extracellular environment. And animal cells, contrast to that, only have one barrier, and that is the phospholipid bilayer, okay? That's a layer just outside the cytoplasm, and every cell has a phospholipid bilayer. The definition of a cell is that they are a protoplasm surrounded by a membrane. A protoplasm is just cytosol and dissolved solute and organelles involved in there. Okay, so every cell, every single cell is surrounded by a phospholipid bilayer or a membrane. It doesn't have to be a phospholipid bilayer per se, but a membrane. Okay, and that is present then in both gram-positive and gram-negative organisms because they're cells. But they have different than surrounding things around that. They have different, let's say, peptidoglycan cell walls around them, and they have different numbers of those members, but every single one has at least one membrane. Like I just told you, both gram-positive and gram-negative bacteria have cell walls. This is the example of the E. coli peptidoglycan cell wall. It's actually a gram-negative bacterium, but it's irrelevant. It's irrelevant because the basic structure of the cell wall is very simple. You have a repeating disaccharide unit. In this case, it's NAG-NAM, 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 linked up by beta-1,4 linkages. And then coming off of some of these sugar residues is going to be then a amino acid side chain. In this case, it's the 4 amino acid side chain. L-ala, D-glu, L-lysine, D-ala, irrelevant again. And then you have a bridge, some amino acid bridge connecting the disaccharide units. In this case, in the case of E. coli, it's the pentaglycine bridge. And that's the basic structure, and this will repeat over and over and over, and that'll make up the peptidoglycan cell wall. Called peptidoglycan because you have peptides and you have sugars, glycan. There are a few things that I want to mention here. One, this is the lysozyme cleavage site. Lysozyme will cleave NAG and NAM residues, which is found in many different bacterium. And so that lysozyme is found within the lysosome, which is an organelle within our body. As you'll come to appreciate, when we phagocytose bacterium, the lysosome will fuse with the endosome to form a phagolysosome, and it will destroy bacteria in part by utilizing lysozyme to destroy the cell wall of the phagocytosed bacterium. The other thing I want to mention here is penicillin. So penicillin is an antibiotic, and it works actually by binding to a protein called transpeptidase, which actually catalyzes the formation of these bridges between the disaccharide units. And of course, then that'll ruin the structural rigidity of the peptidoglycan cell wall and ultimately lead to the death of the bacterium. That's how penicillin works. Now, what we'll move on to then is how the gram-positive and gram-negative bacterium differ just in general, but as well as in the cell walls. So they actually differ in the extent to which the cell wall is present. So here's gram-positive bacteria. What you find here is embedded proteins, a cytoplasmic membrane, and a peptidoglycan cell wall. There's one thing to note about this cell wall. It is huge. It is hugely thick. It is very, very thick. And that's one of these structural identifiers of gram-positive bacteria, this thick peptidoglycan cell wall. Another thing that's important about this structure here, they have cytoplasmic membrane, then peptidoglycan cell wall. The peptidoglycan cell wall does not block the diffusion of low molecular weight compounds. So something like an antibiotic can flow right through that cell wall and get to then the cytoplasmic membrane and cause a lot of problems. Okay. Any other small weight molecules, small molecular weight molecules pass right through that peptidoglycan cell wall. And so gram-positive bacteria have a real big problem with things like penicillin and other antibiotics. And just to remind you again, what is that cell wall? Well, that cell wall is a repeating disaccharide unit with these amino acid side chains that can then be cross-linked by, let's say, transpeptidase. That's what a peptidoglycan cell wall is. And these peptidoglycan cell walls, the gram-positive bacteria have a real big problem with things like lysozyme and things like penicillin, because again, this is not a barrier to their diffusion. Okay, that's gram-positive bacteria. Again, embedded protein, cytoplasmic membrane, and peptidoglycan cell wall. Again, every cell has a cytoplasmic membrane. So every cell we talk about is going to have the cytoplasm and the cytoplasmic membrane, phospholipid bilayer. Now let's move on to gram-negative bacteria. Again, every cell has a cell membrane. So he has embedded protein and a cytoplasmic membrane. Then he has a periplasmic space. Okay, then he has a very, very small cell wall. Again, this is a peptidoglycan cell wall, repeating disaccharide unit with amino acids that can be cross-linked to each other to form a strong structural cross-linked cell wall. But it's very small here. It is not anything like the peptidoglycan cell wall in terms of size that we saw in gram-positive bacteria. So that's one of the distinguishing factors. We have another periplasmic space, and then we have an outer membrane and an outer membrane. Again, this outer membrane is a little bit different than the inner membrane, so we'll go through it. But you also know this thing called murine lipoprotein. Murine lipoprotein serves as an anchor It is a lipoprotein, has both protein and lipid portions to it, and it serves as an anchor to anchor the outer membrane to the peptidoglycan cell wall so it doesn't go flying away. We have porins found in the outer membrane of gram-negative bacteria. So you should appreciate the fact that if we have a hydrophobic layer surrounding the cell wall, that gram-negative bacteria will be resistant to things like penicillin and lysozyme, because penicillin and lysozyme are proteins. They're hydrophilic proteins. They cannot, it is a barrier, the outer membrane is a barrier to the diffusion of lysozyme and penicillin. So gram-negative bacteria are resistant to penicillin and lysozyme. But they do have porins, and porins can allow for things like lysozyme and penicillin, low molecular weight compounds, to pass through them. So gram-negative bacteria aren't on the freeway, but they're much more resistant to those enzymes. You should also appreciate the fact that gram-negative bacteria can randomly mutate the porin protein such that lysozyme or penicillin or both of them can no longer pass through the porin protein. But that's a channel. And the last thing I want to talk about is lipopolysaccharide, or LPS. So LPS is the major cause of septic shock in the ER, and if we have time to talk about it, we will. But the lipid A portion of that is a big cause of septic shock. What happens is when our body lyses the gram-negative bacteria, it releases a lot of lipopolysaccharide, and then that causes then septic shock and bacteremia. But anyway, the big difference between gram-positive and gram-negative is this small cell wall and this double membrane on the gram-negative bacteria, okay, and the periplasmic spaces. And you have the murine lipoproteins, but the periplasmic spaces and the membranes, right? So again, in summary, gram-positive versus gram-negative. Gram-positives have two layers, an inner cytoplasmic membrane and an outer thick peptidoglycan layer. They're vulnerable to attack by lysozyme and penicillin. And gram-negative cells. The three layers include the inner cytoplasmic membrane, the thin peptidoglycan layer, and the outer membrane with lipopolysaccharide. They are resistant to lysozyme and penicillin attack, and they have a murine lipoprotein anchor. They have porin channels in the outer membrane that allows for fluid to flow through them and some low molecular weight compounds. And they have two periplasmic spaces. Those are the distinctions between the two. Know them. So bacteria have four major shapes: cocci, bacilli, spiral, and pleomorphic. So let's go through them. Cocci, or coccus, stands for ball, right? Coccus singular. Here's cocci. And again, they're circular shaped. Bacilli. Again, they're bacillus-shaped, or these rod-shaped organisms. There's another word for them. Spiral, so you can be spiral here, or you can be comma or S-shaped. And we have pleomorphic. Pleomorphic is essentially no really large defined shape, so just random shapes. Okay, and you can have different patterns. You can have like things like diplococci, and you can have things like clustered or strips. But the four main categories are cocci, bacilli, spiral, and pleomorphic. You should also appreciate the fact, and I mentioned this earlier, that organisms are grouped and named according to their shapes and other qualities, like their use of oxygen or lack of oxygen use. So something like Streptococcus pyogens. Streptococcus. So that means it's cocci shaped. Okay. Staphylococcus aureus. That means it's cocci shaped. You got Bacillus anthracis, which is called as anthrax. Okay, those you can tell the shape from the name. So don't, if an MCAT question asks you about the shape of a bacteria, just go look for the name. It's probably in there. And also that'll also indicate things like metabolics, if it has that kind of name or not. So those, again, that's the importance of knowing that, is to know how to apply that. All right. So we'll talk about virulence factors now. This is cool, so you should enjoy this. This is fun. This is what you want to do when you grow up, essentially. So virulence. Virulence is the degree of an organism's pathogenicity, and virulence depends on certain cellular structures and bacterial toxins, which are termed virulence factors. Nothing special about that. Of course, in order for a bacterium to cause disease, it's going to need the factor that cause disease. A bacterium on its own per se may not cause disease, but it may have these toxins or these other certain cellular structure that then can cause disease, and without those structures, they can't cause disease. So they're termed virulence factors, and that includes things like flagella, pili, endospores, toxins, and biofilms. We'll go through all of them, the ones that we mentioned. So we'll move on to another virulence factor known as the flagellum. I'm sure you're familiar with it, and it's important for some bacteria to infect us. The flagellum is the motile structure made of flagellin protein. The flagellin would be this portion of it, okay? What's really important for the motile property of the flagellum is going to be the basal body. The structure within the basal body known as the axoneme, right? And what the axoneme is just a very defined arrangement of microtubules and dynein motors. And it is what produces that movement, that oar-like motion that we see with the flagellum. How it actually does it is not really well understood, how it actually does it, but I can tell you what kind of goes on there. So as you know, dynein motors are motors. They're motor proteins. They use a binding, hydrolysis, and the release of ATP to either produce directed movement or to move other compounds. What happens in the case of the axoneme is that you have these two microtubule doublets, and you have a bunch of these doublets here. They all have dynein motors, and the dynein motors will use the binding hydrolysis and release of ATP to move one of the microtubules upwards and to remove one of them downwards. So this is coming up at you out of the page, and this one's going into the page. And that ultimately produces that oar-like motion. It's not really understood how, but you can imagine then if something's going up here, something's going up here, and something's going down, that would ultimately, well, something's going up here and something's going down here, that would produce an oar-like motion. And that's flagellum. But, you know, the bacteria can actually change the rate at which they do this and how much they do this and in what direction really that they want to go. And how they do that is through the use of binding of chemokines via chemokine receptors, initiating second messenger signaling pathways that lead to then either the upregulation or the downregulation of the dynein motors or the microtubules, and hence the oar-like motion. So chemotaxis, that's moving towards or away from a concentration gradient. We're going to look at that a lot in the context of the immune system, as our immune cells like to do that a lot, but also almost any cell will do chemotaxis given the chance. There's nothing magic about it, it's just ligand binding to receptor, initiating second messenger signaling pathways that lead to then the movement either towards or away from a concentration gradient depending on what compound is being bound. Compounds that can have this chemotaxic effect are known as chemoattractants or chemokines. Okay, pili or fimbriae it's also called. They are straight filaments arising from the cell wall of bacteria. This is very different from cilia. It's not the same thing as cilia. Cilia are filaments that result from actin polymerizing and then cause us for like a wave-like motion that helps us with moving mucus through our body and other mucosal membranes. But cilia is different from pili. Pili don't move. Pili don't move. But they're very often vital to the virility of a pathogen. So let me give you some examples. Many pili serve as adherence factors. For example, Neisseria gonorrhea has pili that allow it to bind to the cervical and buccal cells and called gonorrhea. So that's one example. And then the sex pilus, right? The sex pilus is a special kind of pili used by bacterial gene transfer, which we'll go through later in this lecture. But essentially, it's used then to form a contact, okay, a cellular contact with another cell, and then transfer bacteria to it. That's a sex pilus, a special kind of pilus. Pili plural. Again, if you were to get rid of the pili on some of these bacteria, and this is the pili right here, these yellow guys. If you were to get rid of the pili on some bacteria, they would lose its virulence because it is a virulence factor and is oftentimes vital for a pathogen to cause disease. So like E. coli, if this is E. coli, and if E. coli was to lose its pili, it would no longer be able to cause diarrhea because it can no longer bind to the intestinal tract. But that's pili. Capsule. A capsule is the peptidoglycan layer that protects pathogens from phagocytes, from phagocytosis. How it does that is that, so phagocytosis is receptor mediated. You need a receptor to bind to a moiety on the thing you want to phagocytose in order to get phagocytosis. It's receptor mediated. Binding of the receptor initiates second messenger signaling pathways that ultimately lead to then the engulfment of a pathogen. If you don't have a ligand to bind, you can't get receptor-mediated phagocytosis, okay? Because there's no moieties for then the phagocytic cells to recognize and cause then phagocytosis. But opsonins are used by the body to combat this, to combat the fact that a bacteria has a capsule, because that capsule is really protective, protects the phagocyte very well. And if we didn't have opsonins, we wouldn't be able to combat it. So opsonins are proteins or chemical mediated, really, mostly proteins though, that bind to the capsule, can recognize moieties on the capsule, and then that opsonin is then recognized by a phagocytic cell to initiate receptor-mediated phagocytosis. If you didn't get that, we'll cover it more in immunology. Endospores. Endospore are the metabolically dormant form of the bacteria that are resistant to heat, cold, drying, and chemical agents. I drew the structure here. I don't care that you know that. But you should know that they activate in favorable environmental conditions. So they may not activate when it's really cold, but if they feel that they're at 37 degrees Celsius and they just got into your human body, yeah, they'll activate then. And that's the structure. I don't care that you know it. You have a cell membrane, thick peptidoglycan mesh, another cell membrane, a wall of keratin-like protein, and the exosporum. Again, I don't care if you know that. But that's one of the big reasons why we have to autoclave instruments, is that these endospores are very resistant to heat, cold, drying, and chemical agents. So you need to get very harsh conditions in order to get rid of these endospores. And the last thing we're going to talk about is the biofilm. Again, this is all about virulence factors. A biofilm is an extracellular polysaccharide network similar to a capsule, secreted by some bacteria that serve as the mechanical scaffold around bacteria, physically protecting it from antibiotics. Again, antibiotics are a low molecular weight compound that then usually inhibit some form of bacterial ribosome function. So, but in general, they're antibiotic. They kill biotics or biota, like microbiota. But it's an extracellular polysaccharide network. It's just like a capsule. It protects the bacteria, but in this case, there are moieties that can recognize the biofilm by the phagocytic cell. That it's not that kind of similarity. It's a similarity since it's a polysaccharide network. So let me give you an example. Our skin is covered in microbes. Some are harmless, but about 30% of us have a bug called Staphylococcus epidermis living on our skin. And when we have no cuts, he's harmless. But if you get a cut, or you go to the ER and you have to get an intravascular catheter into you, if your skin was not cleaned properly, this bugger forms biofilms on intravascular catheters and causes bacteremia and sepsis. So it can be very dangerous. So that's what biofilms are all about. They're just like a polysaccharide bunker that protects the bacteria. Again, virulence is the degree of an organism's pathogenicity. There are many virulence factors that then the virulence of the organism depends on these virulence factors. And that's flagella, pili, endospores, toxins, and biofilms, and more. Toxins. We didn't talk about toxins. But there are a bunch of toxins. So you can imagine how a toxin works. Alright, so we'll continue a little bit. Relationships to the host. A symbiosis refers to the relationship between a host and an organism that lives in the host. There are three main types of symbiotic relationships. So this is a symbiotic relationship. We're not defining what kind of symbiotic relationship it is, but all we have is this cell, this bacterial cell, and this is its chromosomes, and then we have these little other microbes living within that bacteria. Little smiley face guys. So there's a commensal relationship. For example, one organism benefits at no expense to the other. So this is the whale and barnacles. So the whale doesn't get anything from barnacles being on its skin, but the barnacles get a lot from they have protection by the whale. They get food, okay? So that's a commensal relationship. One organism benefits at no expense to the other. The barnacles, these little white things. Mutualistic is both organisms benefit. So here's the cow. So cows have a very mutualistic relationship with the gut microbiota, as do we. So the gut microbiota in cows allow for the cow to eat grass, and that the gut microbiota can degrade the grass, can digest the grass, and then the nutrients that come from the digestion go both to the microbiota and the cow itself. And so in a mutualistic relationship, a mutualistic symbiosis, both organisms benefit. And then there's parasitic, where one organism benefits while hurting the other organism. So in this case, this is malaria infecting red blood cells. They infect the red blood cell, they replicate, and then they kill the red blood cell, unfortunately. And so that's a parasitic relationship. One organism benefits while hurting the other organism. And again, symbiosis refers to the relationship between a host and an organism that lived in the host. Now we'll go through more classifications of bacteria. We talked about gram-positive, gram-negatives. Now we have to go through metabolic classification. So there are three major enzymes that organisms may have at their disposal to rid themselves of reactive oxygen species, or ROS. That's a general term for different types of oxygen species that can be radical species, okay? There's catalase, peroxidase, and superoxide dismutase. So let's go through each one. Catalase. And catalase converts hydrogen peroxide into water and oxygen. Less damaging form of oxygen, right? So this is actually radical, right? So what's the oxidation state on this hydrogen peroxide, by the way? It's -1. It's -1. Check it for yourself. Anyway, peroxidase does a similar thing, and that's found in the peroxisome. And there's superoxide dismutase, which converts superoxide ion into water and oxygen. Good. Those are all good things. Because again, radicals are very dangerous. Radicals can bind to almost anything. They can initiate. So the radical mechanism is initiation, propagation, and termination. So initiation is going to form a radical, maybe by sunlight, breaking a bond or breaking oxygen into these superoxide ions, which can then go ahead and bind to other proteins and convert them into radicals. And that keeps going. And so that's why it's dangerous. So that's why organisms have to get rid of and deal with oxygen appropriately. So there are different organisms and they have different distributions of these enzymes. And that's what call them, in conjunction with other metabolic things, the type of bacteria that they are. So let's go through them. Obligate aerobes. These bugs use glycolysis, the citric acid cycle, and electron transport chain with oxygen as the final electron acceptor. They have all the aforementioned enzymes, so they can deal with oxygen. They can live in oxygen without having any problems. There are facultative anaerobes, and these bugs have the faculty to be anaerobic. Okay, that's where the name comes from. Don't be confused and think that they are strictly anaerobic. No, they can be anaerobic if they want to, but they prefer to be aerobed. In the presence of oxygen, they will act as aerobes. They have catalyzed and superoxide dismutase to deal with oxygen, and they use fermentation for energy. Microaerophilic bacteria are bacteria that can only tolerate low amounts of oxygen because they only have superoxide dismutase, and they use fermentation for energy. So fermentation is glycolysis, and then the conversion then of pyruvate into, let's say, lactic acid if we're talking humans, or alcohol if we're talking other bacteria or some other compound. So that's fermentation cycle. Fermentation the glycolytic cycle refers to then that cycle. That cycle of going through glycolysis, getting to pyruvate, converting that to, let's say, lactic acid, and then getting those NAD pluses so that we can then go through glycolysis again. And obligate anaerobes. Obligate anaerobes are bacteria that have no enzymes to defend against the free radical generated by oxygen. They will die in the presence of oxygen. Those are the guys that die. Those are the guys that don't have any of these enzymes and die in the presence of oxygen. And they die in the presence of oxygen because they don't have these enzymes to combat against free radical oxygen species like hydrogen peroxide and superoxide ion, or reactive oxygen species. So those are just other classifications. I don't expect you to know any of that. Just appreciate the fact that there are bacteria or bugs that can only live either without oxygen or with oxygen. Okay. All right, metabolic classifications continue. Phototroph. It uses light as an energy source. Don't have much to say. Chemotroph uses chemical compounds as an energy source. You can subdivide that into autotrophs, which uses inorganic chemical sources such as nitrogen and sulfur containing compounds as an energy source. Those are called diazotrophs if you're going to use nitrogen. And heterotrophs use organic carbon. So organic means carbon containing. All right, prokaryotes, bacterial reproduction and gene transfer. So here's binary fission. Very simple. You take the bacterium, it replicates its DNA, and then it splits. And then you get another bacterium. So now you initially started with one bacterium, and now you have two. Now you can undergo binary fission again. And then you end up with 4. Again, we just double the amount that we had initially. We go binary fission again. Now we have 8. Do binary fission again. Now we have 16. We keep doing that, we keep doing that, we keep doing that. You might have noticed something. The growth of bacteria is exponential. It's exponential. Okay, every time you do a binary fission event or a reproduction event, you double the number that you originally had. So it's exponential. So if you were to plot bacterial growth over time, or the number of bacteria over time, it would be an exponential curve, which we'll see later on. But that's binary fission. Okay, it's very simple. You don't have to know any of the molecular steps in between getting from this step down to this. We have budding. Okay, there's 2 ones I'm going to talk about: bacterial fission and budding. This is budding, and this is a hydra budding with 2 buds on it. So a bud is essentially just a smaller version of the organism with all the appropriate DNA and proteins, whatever, that then is going to grow into a big hydra over time. All right. So here the concept checked. If you start with 1 bacterium, how many bacteria do you expect to have after 6 binary fission events? I told you it's exponential. So then the equation you should use is probably going to be exponential. So the answer is 64 bacteria. You should have paused the video and done that. If you didn't, well, whatever. Note the trend. Bacterial growth is exponential. To calculate the growth of bacteria via binary fission, you use the equation 2n, 2 to the n, where n is the number of fission events. That's the same kind of equation you use to calculate the number of DNA that would result from, let's say, PCR or DNA replication. 2 to the n. Simple math. Because again, imagine you have one fission event, then you have 2 bacteria. You have 2 fission events, then you have 4 bacteria. You have 3, then you have 8 bacteria, okay, etc., etc. So that there was asexual reproduction, okay? Asexual. It doesn't require a partner. Now, what we'll go through next is essentially called sexual reproduction, and I kind of find it odd because there's no reproduction occurring yet. It's just a transfer of genes, so I call it gene transfer. But this is what we're going to go through next is sexual reproduction. It requires the partner of some sort or form. So here's bacterial gene transfer. There's 3 methods of gene transfer in bacteria: transformation, conjugation, and transduction. We will go through all 3 in detail. Let's start with transformation. Transformation is the addition of DNA via direct uptake of exogenous DNA from the environment. This can be, let's say, in a laboratory, or it can be in just some random environment that the bacteria is in. So what's going to happen is the bacteria is going to get heat shocked. So you're going to increase the temperature of the environment. And that's going to cause for then the activation of sigma proteins associated with heat shock, which will then bind to promoter regions associated with heat shock proteins, and it's going to express proteins that then increase the permeability of the membrane. It's going to make the membrane holy, and that's going to allow for a plasmid to enter. All right, and then when we cool the bacteria down, the membrane becomes less holy. And now what you've done then is you've transformed the DNA. You've added a plasmid DNA into then the bacteria. Some of the things that might arise when you're watching this is what happens to the cell wall. Well, I told you, the cell wall, the peptidoglycan cell wall, is not a barrier to the diffusion of low molecular weight compound. The plasmid is very small, so that's not a barrier to it. So what you need to get is holes in the cell membrane, which we did. All right, so that's what we did, and that's just all that did is add exogenous DNA into the cytoplasm. And now, so long as they have the promoter region, it can then be expressed within the cell. So that was any random transformation. But let's say we're in the laboratory and we had this plasmid. Most plasmids have to have an origin, an antibiotic resistance gene, and then whatever insert you want to put in. All right, it has to have a promoter region too. And again, because you want the genes to be expressed, so that's why you need the promoter region. You need the origin because it's the origin of replication. It is where the DNA will be replicated. If you don't replicate a plasmid, it's just going to die off. You're not going to have enough copies to make enough protein to even have an effect on the cell. That's why you need all those things. What we do then is we transform the plasmid into the bacteria. So we heat shock the bacteria just like we did here, and the plasmid gets taken up, and now it can express our protein. All right, and it can make copies of our protein. And the promoter on the plasmid is such that it makes a lot of copies of it. The promoter, the very strong promoter. We use the very strong promoter on our plasmid so we get many copies of our insert, or of our plasmid, and many copies of the protein that is expressed from our plasmid. Anyway, and that plasmid also has an antibiotic resistance gene. So that allows us to differentiate between cells that haven't taken up our plasmid and cells that have. Because cells that have will not grow on an LB antibiotic plate. So let's say if it's penicillin, then we're going to use a plate that has penicillin on it, so then any cell that took up our plasmid has penicillin resistant, and that can survive the penicillin, and so that's how we know that that cell took up our plasmid, unless that cell was contaminated with something else, but that's a different story. Anyway, that's experimental stuff. That's transformation, okay? It's the addition of DNA via direct uptake of exogenous DNA from the environment. So question, you are a researcher and you want to add another insert to your plasmid. Which restriction enzyme should you use? So a restriction enzyme is a sequence and structural specific enzyme that will cut DNA at a certain sequence. And these are the sequences involved here. So which place should we cut to add a new insert? Assuming that wherever you put it, there's going to be a promoter for that insert. So the answer is EcoR1, this guy right here. Because this cut will not affect the antibiotic resistance gene like Claw1 would, and it won't affect the origin the way BCE1 would. Because BCE1, you'll cut this, and it'll cut out the origin. If you use Claw1, you'll cut out a piece of the antibiotic resistance gene, and obviously, this is a sequence of DNA. If you mess up the sequence of DNA by inserting an insert there, you're not going to get an antibiotic resistance gene. It's going to be mutated. And EcoR1 does none of those things. EcoR1 just cuts the plasmid at some random spot and inserts the insert, and then you can insert your insert that way. You ligate it together. So EcoR1. All right, conjugation. It's the transfer of genetic material between bacterial cells via a sex pilus. You should appreciate the fact and understand the fact that if you're going to transfer things through a pilus or like a column, you're going to need cell-cell contact. You need that. That's conjugation. It's a transfer of genetic material between bacterial cells via a sex pilus, or via this column that allows DNA to transfer. So you should, to understand that, you need to understand that bacteria exists in 2 forms, F plus and F minus. Okay, F plus contains 2 subforms, HFR and F prime. So the plus and minus just refer to the presence or absence of the F plasmid. The F plasmid is a plasmid, a very small plasmid that contains the genes associated with conjugation, or the formation of the sex pilus, the pilus genes. If you're F+, you can form a sex pilus. If you're F-, you can't. Matings only occur between F+ and F-. Okay, so the F- is considered the female, and the F+ is considered the male. The pilus is very much like a penis. Okay, it's different from human genetics, though. So again, F+ contains the pilus genes. HFR, or high-frequency recombinant cells, are a form of F+ cells, but they're different in the sense that they incorporate the F plasmid into their genome via homologous transposable elements. That has consequences. That means then that when they go and transfer the F plasmid, they're going to take some chromosomal genes with it. And when you do that, you're going to have a lot of recombination events, because there's going to be sequence homology between the chromosomal gene that you're transferring to and the recipient cell that's going to also have homologous genes associated with the chromosome. So you'll see what I mean by that in a second. And there's also F' bacteria. And F' bacteria are bacteria who were HFR, but then they looped out a portion of the F plasmid again, containing the sex pilus genes, and now possibly other genes unrelated to sex genetics. And so that's an interesting too, and that also then leads to a lot of recombination events with the genes that were from the chromosome. Okay, so you'll see that in a second. Again, matings only occur between F- and some other form, be it F+, HFR, F'. This is all about the transfer of genetic material. Bacteria are only going to do this when it's favorable to do it. When they need to adapt, for example. That's one of the differentiations between when are we going to use sexual and when we're going to use asexual, right? If the bacteria have already adapted and they're good, asexual production is what they're going to use. If they need to adapt, then they probably would use then conjugation, some other bacterial gene transfer. Anyway, let's get back to conjugation. Alright, so why don't we conjugate some bacteria? Let's do F+ to F-. So the first step is the sex pilus has to extend and bind to the F- cell, from the F+ to the F-. Alright? Once they get drawn up together, it'll contract, it'll get themselves real close, and a passageway will be formed, and now you can pass DNA through that passage. What'll happen is there'll be a DNA that'll come in and cleave this plasmid. And the reason why we need to do that is because we don't want to give up our entire plasmid. We don't want to do that. The F+ cell wants to keep its plasmid. So what it does is it makes a little cut, okay, and it transfers one strand, a single strand of the plasmid to the F- cell. This is a very small plasmid, by the way. It's very small and takes a very short amount of time. And let me remind you that there are no genes on this F plasmid except for the genes associated with conjugation. That is it. So it's very small. So what happens is we cut it with the DNase, we send over a complementary strand, and then polymerase will come in and remake the complementary strand such that both the cells at the end of F+, F- conjugation are F+. Alright? Again, it is very important for you to know that the only thing on these plasmids are going to be the genes associated with the formation of the sex pilus and conjugation. That is it. Where we're going to go to next is not going to be the case, and so that's why I'm making that distinction now. So where we're going with it is the HFR cell, or the high frequency recombinant cells. In order to understand what that means, we first have to understand how we get an HFR cell, and then we'll look at the transfer, and then finally we'll understand why they're called high frequency recombinant cells. The formation of an HFR cell is actually a very rare event. Very rarely, an F plasmid will have one of its insertion sequences that have sequence homology with another insertion sequence, and will allow for a crossover event to occur. Notice that this is the crossover here, and what's going to happen is going to incorporate into the chromosome of the HFR cell. It's now found within that circular chromosome of the haploid cell, and it's now called then an HFR cell. So all an HFR cell is a cell who has incorporated the F plasmid into their genome. Again, an HFR cell, what they are is a cell that has incorporated the F plasmid into their genome. The only way that it can do that if it has an insertion sequence here, and insertion sequence is just transposable elements that allow for crossover insertions or deletions to occur, and a matching one here. So it has to have sequence homology here in order to get this to occur. The transposable elements are genes that can jump from one locus to another. And that can happen in many different places on the chromosome of the bacterium. So it can insert between, say, gene C and gene D using this insertion sequence, or it can insert between E and F using this insertion sequence, right? So you can have many different HFR cells that result and then actually transfer different genes to an F- cell. That's what we'll go through next, and that's really the ramification of an HFR cell, that you can get what's called a merodiploid or a partial diploid, and I'll show you that on the next slide. There's also another ramification, and that is that you'll really never get to transfer the entire plasmid. So we'll go there next. But note the trend: recombination requires sequence homology. So here we are with the HFR F- cross. Again, the HFR is just a cell that has incorporated the F plasmid into the chromosome. Now, I know that I made the chromosomal DNA look very small here, but it's not. It's actually very large. This is the entire genome we're talking about. This is not the same size as the plasmid, and that is going to be critical for your understanding of why an F- cell does not become HFR once you've started the transfer between an HFR and an F-. So the sex, everything normal in the beginning, the sex pills extend and binds to the F- cell wall. They contract and a passageway is formed. A DNase will cut the HFR cell and begin to transfer DNA. However, here's the issue, and here's the issue. The F plasmid, right, what happens is you make a cut and you start to begin to transfer the plasmid. And in the F plus to F minus, the plasmid was really small. So you can transfer everything very quickly. However, in this case, you cut the plasmid, and now you have to transfer the part of the F plasmid that you cut, and then the entire genome, and then the other portion of the F plasmid that's left there. You can see that would be a problem very quickly because that would take a tremendous amount of time. And the reality of it is it would never get there. Okay? It would never get there. What will happen is you get some physical vibration that may break up the cells over time, and then what will happen is you'll get a part of it transferred and a part of the chromosomal DNA transferred, but not the full thing. But there's the ramification there. There's in fact the ramification there, because the reality of it is you've just transferred some genomic DNA, right? And that genomic DNA may contain, let's say, gene A. And that gene A may have sequence homology with gene A from the other cell, right? And now what you have is the marrow diploid, a partially diploid bacterium having one gene on its own chromosome and one gene introduced by transformation, conjugation, or transduction. It's a partial diploid, but it can only be that way for a short amount of time, because the reality of it is the bacterial cells recognize uncircularized DNA or linear DNA as foreign. They have an immune system, and that immune system will recognize the linear DNA as foreign and be degraded by exonucleases and endonucleases. The reason why they're called HFR cells, the high frequency recombinant cells, is that there's sequence homology between these two little buggers here. So guess what? They can undergo recombinations before this happens, before the degradation of the DNA happens. And that can fix, let's say, for example, if this guy had a mutation, it was A-, and this guy is A+, then they can do a recombination event to make the chromosomal DNA A+, and this little DNA that's going to get destroyed A-. Wouldn't that be a great thing? And that's what some cells do, and that's why they're called high-frequency recombinants, because there's so much sequence homology that they can do recombination events over and over and over. So that's why. And this is really the reason why I talked about HFR cross F-, just to talk about marrow diploids. And to remind you of the fact that most bacterium are haploid. So here's the F' cell. What the F' cell is it was an HFR cell who looped back out the F factor into a plasmid. And so it took all of the, the F factor plasmid, but it also took some genomic DNA with it. How it does that is actually a recombination event, a very rare recombination event, where it had limited sequence homology and then does a recombination event that leads to the circularization and the removal of the F factor from the genome of the cell. The point of the matter is that it brings genomic DNA with it, and once it goes to cross with an F- cell, it can actually transfer both the F factor and genomic DNA with it. And that can have a number of ramifications. As you know, as I said earlier, that the bacterium are haploid. All bacteria are haploid. And so when you transfer that, you can actually get a marrow diploid or a partial diploid. And it actually be permanent, right? This will be permanent because the circularized is not recognized by the immune system of the bacterium. And so if, let's say, this bacterium had a LAC-I-, you can actually fix the phenotype by bringing in a LAC-I+ gene via an F' plasmid. So here's a summary of bacterial conjugation. If you cross F+ to F-, most cells will become F+, and recombination occurs at low frequency. And you can never get a marrow diploid or a partial diploid via F+ F- cross, because the only thing on the F plasmid for F+ cells is the F plasmid. There are 6 genes. If you cross HFR cross F-, most F- cells do not become HFR because it takes too long to transfer. Recombination occurs at high frequency for the chromosomal genes close to where the F factor is integrated and in the proper orientation. The rest of the transferred DNA is chopped up by by exonucleases and endonucleases in the cytoplasm of the bacteria, because the bacteria don't have single-stranded or non-circular DNA. So that's a recognition site for them, the enzyme that they have to protect themselves from invading pathogens of their own. And then we have F prime cross F minus, and most F minus become F prime. Why? Well, recombination occurs at high frequency for the sequences on the F prime plasmid, but at low frequency for any other bacterial genes. And why did that happen? Well, because, again, the F prime, the cell, the recipient F minus cell does not have an F prime plasmid. So it can only undergo recombination with sequences that he has homology to. The only thing he has homology to is the gene that came from the chromosome of the donor cell. You gotta think about that. But that's bacterial conjugation. The whole reason we went through that is so you can understand the notion of marrow diploid and partial diploids and how that can result in bacteria, which are normally just haploid. So we will move on then to transduction. Transduction is the process by which foreign DNA is introduced by a bacteriophage into another bacterium. A bacteriophage is a virus that infects bacteria. What we'll need to do to understand this process is go through the viral life cycle, the generalized viral life cycles, that is the lytic and the lysogenic cycle. These are cycles that most viruses go through. We'll particularly look at the phage, lytic, and lysogenic cycle, and then we can finally understand the transduction process. So the first step is that the phage has to attach onto the host and then subsequently inject its DNA. This DNA will be injected, and it will be linear at first, and if it's not degraded by the endo and exonucleases found in the cytoplasm of the bacteria, part of their normal immune defenses, then it will circularize and no longer be targeted as foreign. Then what that will allow to happen is the DNA replication, transcription, and translation of this phage DNA to form new virons. Okay? These phage virons, these protein coats, the protein coats will be translated and will have to form up with one another. And they will surround phage DNA. And by the way, virons are just viruses that have resulted from the lytic cycle. Eventually, these cells will lyse due to the amount of virons within the cell, and now those virons can go on and infect new cells and inject more of their phage DNA into the cell. Well, here's what happens in transduction. Sometimes right here at this process, the phage will package up bacterial DNA into its capsule with its viral DNA. When it goes to infect another cell, it may introduce the old bacterial DNA into the genome of the new host. That process is known as transduction. Now again, I told you that the phage DNA will circularize and then get transcripted and translated and go out and form new virons and go out and infect new cells, but that's not the only type of cycle we can get. And so you can actually get the incorporation of that old bacterial DNA into the genome of the new host, which will then be permanent. So right, we went through the lytic cycle. The two cycles, the lytic and the lysogenic. The lytic is as follows, right? We already went through it. Lysogenic, though, is something we haven't gone through yet. So again, the phage attaches to the host cell and injects its DNA. Phage DNA circularizes, and then the environmental factors determine if we go through the lytic or lysogenic cycle. In the lysogenic cycle, the phage DNA will actually integrate itself into the bacterial genome, becoming a prophage. Again, if the phage DNA that was injected into the cell contained genomic DNA from an old bacterium, then guess what? You just incorporated genomic DNA into the new bacterium. And anyway, in this prophage phase, the virus is actually dormant. So the cell, it'll go about its usual business, producing daughter cells with viral DNA. After a while, okay, some environmental factor will induce the entry into the lytic cycle, right? Some chemical in the surrounding will bind to a receptor and initiate second messenger signaling pathway that leads to, let's say, an activated protein to bind to an activator element in the promoter region of this locus, and will induce then the transcription and translation of the viral DNA and entry into the lytic cycle. But again, the transduction process is really right here, right? One thing I forgot to mention about transduction is that when you can do some studies where a virus transduces DNA from one bacteria to another, and if you see that two genes are always transduced together, then you know that they're very close together on the chromosome. Because they have to be, because you only, the virus is very small. We already went through that. The viruses are very small compared to the bacterium. So they can only fit so much DNA within this capsid. So if you keep finding two genes together in every time you do a transduction between a virus and another bacterium, then you know those genes are very close together on the chromosome. There are 3 methods of gene transfer. Again, transformation, which is genetic addition of DNA via direct uptake of exogenous DNA from the environment. Conjugation, which is the transfer of genetic material between bacterial cells via sex pilus. And transduction, foreign DNA is introduced by a bacteriophage, a virus that infects bacteria. So now we'll continue with bacterial growth and gene expression. I mentioned bacterial growth before, and I said it was exponential, which it is. But, so here is a graph or a figure from Monod, Jacob Monod, if you're familiar with that name. He's the guy who talks about lac operons and all that jazz. He saw that so on the x-axis you have time, on the y-axis you have log cell number. He saw that the curve was exponential when all he added was glucose to a plate of, let's say, E. coli cells, or bacteria, don't matter, bacteria, E. coli. And when he did glucose and lactose, so 50% glucose and 50% lactose, he saw 2 growth phases. He saw that there was exponential growth, and then it flatlined a little bit, and then it picked up again. It's called dioxy, meaning 2 growth phases. Does anyone know why there are 2 growth phases? Well, why is what we're going to go through next. So I think it's going to be instructive, and it's going to allow you to practice your thinking skills and your problem solving skills, and your comfortability with genetics. So here's the lac operon. The lac operon is an inducible system, and it has points of both positive and negative regulation. So I'm just going to name each thing here. So let's start on this side. So here are the promoter and the operator region for the lac-I protein. This is a different promoter. This is not part of the lac operon. I'm including it here because it plays a role in the lac operon, but it's not part of the lac operon. The lac-I protein, or the repressor protein, has a different promoter than the lac operon, and that allows it to then regulate the lac operon. Okay, CRP site. What's a CRP site all about? That's an activator site. It is the site where if a protein binds there, it's going to activate the transcription of the locus in question, which in this case is the lac operon. This operon right here. This whole thing from here to here is the lac operon. From this gray promoter region to here is the lac operon. Anyway, the CRP site stands for catabolite response protein, or catabolite activator protein, CAP. It's a different name for it. Again, I told you what it does. LAC-Z is beta galactosidase, and that cleaves lactose into glucose and galactose. The cell cannot use lactose for glycolysis by itself. It needs to convert it into glucose first. It has to convert it into glucose first. So that's why we need beta galactosidase. That's what it does. It converts lactose into a glucose and galactose molecule. And we have LAC-Y, lactose permease, and that allows lactose to enter the cell. You should appreciate the fact that, again, all cells have a phospholipid bilayer. Lactose is not lipid soluble, so it needs a channel to flow through. In order to get that, you need lac permease. And lac-A is irrelevant. It transfers in acetyl group to lactose, but it does not affect it doesn't affect anything about the lac operon. So it doesn't play a big role here. We're not even going to mention it further than that. So now what I'm going to go through is walk you through this lac operon. Again, the lac operon is an inducible system. So let's go through it. I'm going to tell you now. Actually, no, I'm not. Yeah, yes, I am. So the repressor protein. The repressor protein binds to the operator sequence. That's its binding position. When the repressor protein has bound to the operator sequence, you cannot get the RNA polymerase to bind to the promoter region. It prevents RNA polymerase from binding to the promoter region, as you saw there. Okay? It prevents it from binding. So you can't get expression of this locus if the repressor protein is bind to the operator sequence. But you can induce this system by having lactose. So under conditions of high glucose and low lactose, you're not going to have the repressor bound by anything, so it's going to block the transcription of this operon. However, if you get lactose into the system, so let's say you're in conditions of high glucose and high lactose. Lactose is going to get into the cell via lac permease and bind to the repressor protein and then relieve the inhibition due to the repressor protein on the lac operon. However, that is not enough, not sufficient to get high expression on the lac operon. The reason for that is you also need activation of the lac operon via the CRP, or the catabolite response protein, or the catabolite activator protein. You only get that in low levels of glucose, because the stimulus for the activation of the CRP protein is going to be cyclic AMP, well, low level of glucose. So you have low level of glucose that initiate second messenger signaling pathway that lead to then the conversion of ATP into cyclic AMP via adenylate cyclase. Cyclic AMP can bind to the CRP protein and activate it. That allows for the CRP protein to bind to the CRP site. All right, and now you can get high levels of expression. The sigma protein can bind to the promoter region, okay? If it's going to be E. coli, it's going to be a TATA sequence. And then RNA polymerase can transcribe the DNA into RNA. And here the RNA, we know that E. coli cells as well as bacterial cells have polycistronic mRNA. So each one of these have its own Shine-Dark-Arnold sequence that the ribosome small subunit can recognize as well as its own AUG. So what happens is the ribosomal small subunit recognizes the Shine-Dark-Arnold sequence and positions the P site in the AUG, and that positions the P site of the small subunit with the AUG start codon. Then you get F-methynyl tRNA IF2 GTP complex binding to the AUG sequence within the P site. And then that allows then for the large subunit, ribosomal subunit, to bind. That's going to then cause for hydrolysis of GTP off the IF2, and now we're going to be in elongation mode, and that allows for then aminoacyl tRNA EF2 GTP complex to bind at the A site. That binding will then cause then a conformational change such that the F2 will hydrolyze GTP. That conformational change associated with the hydrolysis of GTP will then cause for the peptidyl transferase reaction to occur at the peptidyl transferase center. That then allows for peptide formation. We go through those cycle, then leave at the E site, enter at the A site, and peptidyl transferase occurs between the A and the P site. You go through that cycle, and then that's how you make polypeptides. And eventually you get termination, which occurs when we reach a stop codon, because there are no tRNA that can read a stop codon. Anyway, and these are the proteins you get. You get beta-galactosidase, lac permease, and lac A. Again, that's lac operon. You need two things to get high levels of expression. You need to inhibit the inhibitor, inhibit the repressed protein to induce the system, as well as low levels of glucose, so that you can get CRP binding to the CRP site and activating the transcription and translation of the lac operon, the locus associated with the lac permease and beta-galactosidase. That then the glucose then the cells can then feed on the lactose. So before we move on, I want to mention two things. This first is the notion of an allosteric shift. And the second is this lac permease. I want to talk about where this lac permease came from in the cell membrane. So an allosteric shift, I want to make this clear. An allosteric shift is when the binding of a molecule at one end of another molecule changes the conformation of that molecule at another end. So let me give you an example. The binding of allolactose to the lac-I repressor protein allosterically shifts, called the conformational change in the lac-I protein, such that it can no longer bind to the operator sequence. It ruins the active site of the lac-I protein in respect to binding of the operator sequence, and it can no longer bind. Another example. Alright, so cyclic AMP binding to the cyclic AMP response protein called an allosteric shift. It called the conformational change in the CRP protein such that it can now recognize the CRP site. Okay, so that's an allosteric shift. The binding of a molecule at one end will change the conformation of, let's say, a protein at another end, and that'll allow for a change then in the properties of that protein. Alright, that's allosteric. Now, the second thing I want to talk about is the lac permease. Where did this lac permease come from? Remember, I told you that to get high levels of expression, you need high levels of lactose in the environment that then comes in via the lac permease to then bind to the lac-I protein after being converted to allolactose, and you need low levels of glucose to get the CRP to bind to the CRP site, and you get high levels of expression. So where the hell does lac permease come from? Well, there's a basal low level of expression of this locus, regardless of whether or not lac-I protein is there or not, because every once in a while the lac-I protein comes off. Just from random kinetics, it'll hop off the operator sequence for a second, and that second may just be enough for the RNA polymerase and the sigma protein to get transcription and translation going. Maybe just one little transcript. That transcript is enough to get a lac permease out there to then start to get lactose into the cell to sense the environment. Because otherwise you couldn't sense the environment. You wouldn't get this, because again, if you didn't have a lac permease, you couldn't get lactose into the cell, and then you have a real big problem. But that's that. Now I'll go through it with two different scenarios in a different figure. So we're in the scenario right now that it's induced. So glucose is absent and lactose is present. If glucose is absent, we have cyclic AMP binding to CRP, binding to the cap site. If lactose is present, that means that we're getting lac permease to let lactose in, convert it to allolactose, bind to the lac-I protein, and then we can get high levels of expression. Polymerase can bind, do transcription and translation. Repressed. Let's go through the system where it's repressed. So we're looking at right now, glucose is present and lactose is irrelevant. It's irrelevant because if you have glucose, that means you have no CRP, that means you have no cap, and that means that you don't get high levels of expression, no matter if lac-I is there or lac-I is inhibited. It really is irrelevant because of the fact that you need both those things to get high levels of expression. So that's the like operon. It's an inducible system. I mentioned the notion of positive and negative regulation. I don't really like talking about it, but a negative regulation is where a protein binds and inhibits transcription. So like I, for example. Positive regulation is when a protein binding increases transcription. For example, cyclic AMP protein, well, the CAP CRP complex, the CAP cyclic AMP complex. So those are examples of positive and negative regulation, or negative and positive regulation, respectively. So we're back to this curve. So do you guys know why yet? While it's here, the answer's here for you. When the slope of the curve is the lowest, that is the period when the bacteria are switching from glucose to lactose metabolism. So it takes time to transcribe and translate the proteins that'll then allow for these cells to feed on and grow off the lactose. It takes time. And so that's why we see a dip then in the cell number during this time, because that's the time where they're switching. All right, in this curve, upward curve, only glucose is used. And in this curve, only lactose is used. So that's why we see that dioxy curve, or that double growth curve. So I want to make sure that you understand this and you're comfortable with genetics in general. So we're going to go through different situations here in terms of the genome of some bacteria. So this is the genome of one bacteria where all the genes are present. So the plus means that the genes are present. If you see a minus somewhere, that means the genes are absent. So let's go through it, or they're dysfunctional. So in this genome, in this sequence here, the LAC-I protein is non-functional. It is not present in this guy's genome, in this bacteria's genome, whereas the rest of them are. So the ramifications of this would be, if this guy was haploid, there would be no regulation in terms of the LAC-I protein on this operator sequence. So under low levels of glucose, regardless of whether or not lactose was present or not, you would get high levels of expression of this locus, as long as low glucose levels, because you still have the CRP site, but you don't have the LAC-I protein. How about this guy? In this case, you have actually a C here. So what does the C mean? That means constitutive. So if an operator sequence is constitutive, that means that it cannot be bound by anything. So the LAC-I protein cannot bind here. So although you have a LAC-I protein, you can't bind to the operator sequence. So you're gonna get high levels of expression of LAC-Z, LAC-Y, LAC-A, assuming that you have low levels of glucose in the environment. That's what constitutive means. OC means constitutive expression. And how about LAC-IS? So LAC-IS is the super repressor. It binds the operator sequence, but the inducer cannot bind to it. And I didn't mention this before, but I'm going to mention it now. What happens when allolactose binds to LAC-I protein is that it causes an allosteric shift. So it changes the conformation of a protein at one point such that it can no longer bind to another protein at a different point. That's what allostery is all about, and that's what it does. So a LAC-I superpressor is such that it can bind the operator sequence on one end, but the inducer can't bind to it anymore. And so what happens when you get a super repressor but a no constitutive? So an operator sequence that can't be bound by LAC-I. Well. Even though you got a super repressor, it can't bind to the operator sequence, so you get high levels of expression here, assuming low glucose levels. Okay. So this next conversation is gonna put together what we went through last session in terms of gene transfer. So we're going to take an F prime cell and conjugate it with an F minus cell. Again, an F prime cell has the sex pills genes and some genes from the chromosome. In this case, it had the lac operon genes in the plasmid. So what it's going to do is it's going to transfer itself. It's going to start to transfer. And it transferred the genes. So again, the sex pills gene and the lac operon shortly follows. And then what happens is they replicate, DNA polymerase replicates the single strand that was transferred over. And now you have two F prime cells. And now they both have two. The F prime cell, the recipient F prime cell, has two lac operons now. It has two. Because this F prime cell got the lac operon from its chromosome. So it was lac minus. But this F prime cell that was transferred, this plasmid, has both the sex pills genes now and the lac operon. So now it's a merodiploid, or partial diploid, or a merozygote. And that has consequences. And those consequences you're about to see shortly. So let's say we conjugate this bacteria with this F prime bacteria, and the resulting recipient, what was once F minus, is now this genome. So initially it had this genome, and then it transferred over the F prime plasmid to get this genome. So it's a partial diploid for the lac operon, and we included the operator sequence too, because I know I said lac operon is not part of this, but we always include this. Anyway, the question then becomes, what are the expression levels of these proteins now? Because you had lac-I minus here. So initially, what was the protein? Let's go through the first thing. Initially, you had lac-I minus. So you had no repressor protein to bind to the operator sequence. So on the low level of the glucose, all of these were highly expressed. But when you introduce the conjugation protein, the conjugation genes, you got a LAC-I plus protein. And that LAC-I plus protein can act on both loci to inhibit the transcription of these genes. So let me show you that. So this repressor protein can bind on both the gene, the plasmid that it came from, as well as the chromosomal operator sequence. So it can work in trans. It can bind a locus other than from the one it originated from. That's what it means when something says that it works in trans. It can bind at a sequence other than the one it originated from. Not everything can do that. LAC-Z can't do that. I mean, the operator can't do that. That's an insist. Promoter can't do that. But LAC-I protein can. So factors that work only in cis cannot affect surrounding loci and can only affect its own locus. Factors working in trans can affect surrounding loci. So here's an exercise for you. I'm going to go through the first one for you, and then you can determine... Well, I'm going to go through the second one. Well, the first one. Determine the phenotype for LAC-Z and LAC-Y for each of the organisms listed below, and assume glucose levels are low. So you have a marrow diploid for the LAC-Operon. It's LAC-I plus, promoter plus, operator plus, LAC-Z plus, LAC-Y minus. And then we have for the F prime, the plasmid, LAC-I minus, P plus, operator plus, Z minus, Y plus. So let's look at LAC-Z. What's going to be the expression levels for LAC-Z? Not made, inducible, constitutive. Assuming that low glucose levels are low. Let's see. For Z minus, just looking at this one, you would think that it would not be expressed at all. But you've introduced a Z plus on the well, you have a chromosomal Z plus. So that's going to be inducible for LAC-Z. Y minus, let's look at LAC-Y, or the LAC permease. That's going to be inducible. Again, why? Well, you have Y plus here, but you have no LAC-I, but you have introduced another LAC-I protein via conjugation. So although there's no Y minus on this locus, LAC-I can work in trans. So it can bind to both its locus and another locus. So that's why it's inducible. So you can do the rest by yourself. Here are the answers. I'll give you 5 seconds to pause. 1, 2, 3, 4, 5. Okay. Let's go through a repressible system because this is the MCAT. This is a trip operon or tryptophan operon. So this is the operon in bacteria that are required to synthesize tryptophan. So this is the promoter region and the operator sequence and the structural genes that are required to synthesize tryptophan. Okay, when the low level of the tryptophan, okay, you need to activate the sequence. So this is a repressible system. So TRPR under normal conditions will not bind to the operator sequence when there's no tryptophan in the environment, and you'll get high levels of expression of this locus. However, when there is tryptophan in the environment, tryptophan will bind to TRPR, and that'll allow for binding confluency. That will induce an allosteric change in the tryptophan repressor protein such that it can bind the operator sequence. And it's going to go ahead and do that then. And now, sigma protein RNA polymerase can't bind, so you have low levels of transcription and translation. That's a trip operon. It's a repressible system. All this is working through allostery, most of it in terms of binding of co-repressors or whatever. Anyway, note the trend. Negative regulation is when a factor binds to a locus and prevents transcription. LAC-I, TRPR. Positive regulation is when a factor binding to the locus is required for transcription to occur. That's like the cyclic AMP protein, or cyclic response protein, and any kind of activator proteins. Prokaryotes, genetic adaptability. We're going to move on now from the LAC operon and talk about genetic adaptability. So, the classical experiment to determine whether or not genetic adaptation resulted from a physiological response or to random mutation was called the Luria-Delbruck experiment. And it was as follows. The first hypothesis was that resistance is a physiological response to bactericide, and what you would expect in that kind of hypothesis situation is that as soon as you introduce the bactericide in, let's say, E. coli that are undergoing binary fission, you should get some colonies that have then resistance to bacteria, but you shouldn't get that before that time period has occurred. The other hypothesis is that resistance arises from random mutation, and in such a hypothesis, if you take bacteria that are undergoing binary fission, then there should be some colony that die out and don't survive at all, and there should be some that have a huge amount of bacteria within their colony because of the fact that they randomly mutated resistance to the bactericide before the bactericide was introduced. A bactericide is what can kill bacteria. So an antibiotic, for example. So what was true? Well, let's go through it. when they ran the experiment, this is what they got. What they did was they took E. coli that were undergoing binary fission, and then they added a bactericide and quickly then plated them on an agar plate. And they saw which ones grew and which ones didn't. There were some plates that didn't grow at all, and there were some plates that were filled with bacteria. So which one did that match? Which one did that match? Hello, hypothesis B. Resistance arises from random mutation, and that's why there are some plates that have none and some plates that have a ton. And the ones that have a ton just mean that there was a mutation very early in the binary fission process. And so when you introduce the bactericide to the bacteria that you plated onto the plate were already resistant to the bactericide, so you didn't have an issue. Note the trend. Genetic adaptation is the manifestation of random genetic mutation coupled with natural selection. It's not like, I don't like the word adapt because, yeah, the bacteria adapt, but they don't do it on purpose. It's random. It's absolutely random. It's random. Because what happened is they randomly mutate, and then due to the fact that they can survive better now, they can survive better in the environment that they're in, they survive. And the ones that can't survive as well die. And so that's natural selection at work. And they don't do it on purpose. It's random. So we'll finish up with a conversation about viruses and then transposons, and then we'll be done for the day. Let's get into it. So there's some characteristics about viruses that you should know. The first is that they are energyless. They simply drift in space until they come in contact with an appropriate cell that they can infect. Again, they're energyless. One of the characteristics of being alive is having energy and being able to utilize energy and convert energy into different forms. Viruses can't do that. They are not alive. They're also not alive, not considered to be alive, because they cannot replicate their own genome. They cannot replicate their DNA. They cannot survive without infecting another cell. They can survive, but they can't replicate or form new genes, cannot replicate their DNA without using somebody else's materials or enzymes. So they're not alive. Viruses have a protein coat surrounding their genetic material, and some are further enclosed by an external phospholipid bilayer, which may or may not have glycoproteins. While some viruses may carry some structural proteins and enzymes in their capsid, they do not have organelles or ribosomes. They require the use of the ribosomes from other materials and other organisms to survive and replicate their DNA. They do not have any organelles in their capsid. The genetic material of our virus is the DNA or RNA. Again, nucleic acids. It's never both. It's either one or the other. They can be segmented. It could be single-stranded RNA, double-stranded RNA, double-stranded DNA, but it's never both. Replication of the virus occurs when the virus takes control of the host cell's synthetic machinery. Viruses contain all the genetic information, but none of the enzymes needed to build millions of replicas of the original virus. Those replicas, as we saw earlier, are called viroids. So next what we'll do is we'll build up a virus so that you can understand the structure of the virus. So the first thing we're going to deal with is the icosahedral capsid. So viruses come in two forms in terms of structure, two major forms. And that's the icosahedral capsid and then the helical symmetry capsid. So this is the capsimer, this little dot here. And what this is the string of amino acids. And we can fold that up just a globular protein. So we can fold up a capsimer into a globular protein like so. This is just a globular protein like you've always seen before. Just this wrapped up, and maybe more than one of these, and then those can come together to form what's then called it's a triangle. It's a triangle with a bunch of capsimers. And so one of these little circles, and I didn't color them in just to make my life easier, is one of these. So we put a bunch of those, okay? Then you put 20 of those together, and then you form what's called a capsid. So this is a viral capsid. It's an icosahedral 20-sided triangle. Well, 20 triangles put together. And that's an icosahedral capsid. So a 20-face shape. And this protein capsid is what's housing the genetic material of the virus, be it DNA, RNA, etc. Doesn't really matter. This protein capsid is what's protecting that genetic material and housing that genetic material. Now let's go through the helical capsid. What you do is you take the capsimers, like this is the same capsimer we've seen before, and it's RNA this time. You only get RNA helical capsids, and you fold it up to form this helical symmetry capsid. Simple. So that's it. And this is what's surrounding then the DNA and the RNA of the virus. Either it's going to be a helical capsid or a icosahedral capsid. Okay? So those are the two shapes. And then the envelope. So viruses are sometimes surrounded by a phospholipid envelope. So this is how you make it. You take the virus. Again, it's energyless, so randomly it'll start running into the membrane. It'll drift into the membrane. It'll push the membrane out, and the membrane will start to form a figure 8 around it. And then eventually, what happens is there's a hydrophobic interaction between these two portions of the membrane such that you form a circle envelope around the virus, and then these things move in and take its place. So that's envelope formation. Again, you have this protein capsid that then is surrounding the genetic material that is within this capsid. And then some viruses have this phospholipid bilayer envelope. Viruses that don't have this envelope are sometimes called naked or non-enveloped viruses, where the viruses that do have it are called enveloped viruses. Viruses are classified as either RNA or DNA viruses, generally speaking. RNA viruses we'll deal with first, and then DNA. Two types of RNA viruses are the plus and minus strands. Okay, the plus RNA strand means positively ready to be translated. It's a mnemonic. So you have a positive RNA strand, you can translate that straight into proteins. If you have a negative strand, it means here the mnemonic negative, sir. I cannot be translated. If you have a negative RNA strand, you have to transcribe it first to a positive strand, and then you can translate it. But you can't translate it immediately. Okay, so that's what we did there. We transcribed it first. And that's done via an enzyme called RNA-dependent RNA polymerase. And that has to come from a viral genome. Because you know what? We don't have that in our normal genome, so the virus has to come with that. If it doesn't come with that, then it can't transcribe its RNA into its negative RNA into positive RNA. The negative and positive just the names, okay? It's not like it's negative charge and positive charge. RNA has negative charge, period. It's just a nomenclature, okay? So for RNA viruses, I'll give you the example of HIV retrovirus. It's called a retrovirus because it does reverse transcription to DNA to incorporate into the genome. But this is going to be the subject of discussion for adaptive immunity. We'll have a lot to say about HIV there, so what I'm going to do here is just kind of go through this pathway here. So HIV had the phospholipid envelope with glycoproteins associated with it. It had a protein coat, and then it has viral RNA and proteins within that protein coat, such as integrase. What it'll do is it'll enter the cell, particularly the CD4 plus helper T cell, via binding to the GP120 receptor via GP120. It'll initiate endocytosis, it'll get into the cell, it'll decode, and you'll get the reverse transcription of the RNA back into DNA, and then you get DNA replication to get the complementary strand of that DNA, and then you'll get translocation into the nucleus via the nuclear pore complex, all right? And then integrase will make a cut in the genome of our CD4 plus helper T cells and integrate the viral DNA into the genome. And it'll now be a provirus. And then at some point later in the game, you know, some environmental effect will cause for then the activation of that locus and increase the transcription and subsequent translation of the RNA. And then that RNA will be transcribed to either the protein that serves as a capsid or integrase or whatever it needs to then form more virons and then go out and infect more cells. The problem really arises with HIV is what happens to the CD4 plus helper T cell, that they are recognized as the cell and then killed off by the CD8 plus cytotoxic T cells. So anyway, we'll get there when we get there. Okay, viruses are classified as either RNA or DNA viruses. We'll go through DNA now. So here's where it gets a little bit confusing. So DNA strands have that positive and negative nomenclature as well. So in a DNA virus, the positive strand is ignored. All right, and the negative strand is transcribed. So that's what we show here. This is a negative DNA strand, and we have a RNA polymerase transcribing the RNA. And then that can serve then as a site for ribosomal translation, and then that's how you get proteins. So in this case, again, the negative strand is transcribed, whereas in RNA viruses, the negative strand had to be converted into then a positive strand for you to get then translation of that strand. But in DNA viruses, the negative strand is actually the one that's a big player. It gets transcribed into RNA, and then it gets translated. So just kind of going back to earlier in the lecture, I just want to make another size comparison here. What you got here, the human hepatocyte, a liver cell, okay? You have an E. coli within that, and then you get the T4 bacteriophage. The human hepatocyte is so huge that you don't even see the end of it. Whereas we got the E. coli and then the T4 bacteriophage, and you actually have a ribosome here, so that really kind of tells you, you know, the kind of size, relative size going on here. Anyway, we're going to talk about the T4 bacteriophage. Well, bacteriophages in general. So here's the structure of the bacteriophage, and again, a bacteriophage is just a virus that infects bacteria. You got the DNA inside the icosahedral head. You got a neck region, which really isn't shown too well. It's actually behind this little head region. You got a sheath, you got a base plate, and you got pins that will then cause the hole in the membrane of its target cell, of its target bacteria, and inject its DNA through the sheath, through the hole in the base plate, and into the cell. You got two life cycles for the bacteriophage. It's either the lytic cycle or the lysogenic cycle. What happens is the phage will attach onto the host cell. That's the first thing that needs to happen. Then you get injection of the DNA. The DNA will circulate if it's not degraded by the immune system of the bacteria, and then the environmental factors will determine if it goes into the lytic or the lysogenic cycle. If it's going to go through the lytic cycle, what you're going to get is replication of the phage DNA, and then the transcription and translation of viral proteins to form new phage virons, or just new viruses. Okay, you'll get enough, and they'll eventually lyse the cell, and then now those new virons can go over and infect new cells. Okay, if it goes to the lysogenic cycle, what will happen is you'll get the incorporation of the phage DNA into the bacterial genome, and that is called the prophage now, or provirus. And then it'll go out its usual business. Some will replicate, and you'll get daughter cells all containing the viral DNA. And then some environmental factor, just some environmental factor will increase the transcription and subsequent translation of this locus, and you'll get into the lytic cycle. And, let's be clear about something. What do I mean by the environment inducing entry into the lytic cycle? What I mean is there's some chemical in the environment, some molecule that will bind to a receptor and induce second messenger signaling pathways that will lead to the activation of, let's say, an activator protein that will bind to an activator element in the promoter region of the locus in question and increase then the transcription and translation of the locus. And that's the basic viral cycles, the lytic or the lysogenic. So transposon to where we're going to go next. So a transposon, or transposable element, are sequences of DNA or RNA that can be moved from one genome to the next. There are so-called autonomous transposons and non-autonomous transposons. Autonomous transposons contain all the enzymes required to move from one gene to the next. Non-autonomous transposons are transposons that have lost the enzymes required to move from one gene to the next, but could have they been introduced to an environment or a genome where those enzymes already existed. Okay, but in general, transposons are DNA or RNA sequences that can move from one genome to the next. So in order for you to understand this, what I'm going to do is I'm going to go through the mechanism one transposable element mechanism, and that is that of the insertion sequences that we talked about earlier. So here's what happens. The transposinase enzyme that recognizes inverted repeats and excises the transposon, and it has a transposinase gene right here. So it makes cleaves right here. It recognizes these sequences, its inverted repeat sequences, and cleaves. And this is in the genome, let's say in the bacterial chromosomal genome, cleaves. And then now you have this free thing that can move. It can move from one place to another. And then you get cleavage somewhere else in the genome. Somewhere else in the genome, you get an endonuclease to go ahead and cleave the DNA. Because remember, DNA is just a big old molecule. Sometimes we forget that. Sometimes we forget it's just a bunch of phosphodiester bonds with nucleotide bases. Okay, that's all it is. And so you can cleave that. So that's what happens. You get an endonuclease to cleave that. All right, now you can move that transposinase gene in. Now what you need to do is you need to fill in what's missing. How do you do that? You take DNA polymerase, you fill it in. Again, DNA polymerase reads 3' to 5', extends 5' to 3'. So it's going to extend this way. Okay, and it's going to extend this way. Okay. Now what we need to do is we need to ligate these two guys together. How do you do that? DNA polymerase can't do that. Because, well, I'll let you answer the question later. So nicks are fixed by DNA ligase. This is the nick. It is a hole between a 3' and a 5' end. Okay? There's no template in between the 3' and the 5', so DNA polymerase can't fix that. DNA ligase can fix that, and it uses ATP to do that. So it fixes it. So now what you did is you connected two genes that were before not connected, or two sequences that were before not connected. That's what a transposon does. It cleaves itself out of one genome and it puts itself into another genome. And it does that this is one example of how it does it. So the question then is, why can't DNA polymerase fix the nicks? And here's the question answer choices. It only extends 5' to 3'. The nicks are way too bulky. DNA polymerase requires a template strand, or it is DNA ligase's job to fix nicks. Why can't it do it? The answer is C. There's no template here. Watch, let me go back. There's no template here. You have a 5' end and a 3' end. You can't add another nucleotide. You can't add another nucleotide because there's no template. Otherwise, and even if you could add another nucleotide, it wouldn't fit. It'd be an insertion. There'd be no base pair for it to base pair with. There's no space here. So that's why DNA polymerase cannot fix this. They can't fix nicks. Okay. Hope that makes sense. If not, hit me up. That's it.